Very good morning. Good morning, students, participants. Very good morning. Sir, கேக்குதா? இல்ல, இன்னொன்னு ரெண்டு. முடிஞ்சதுக்கு அப்புறம். Shall we begin the session?
அங்க ஆஃப் பண்ணலாம் இல்லை, நீங்க இங்க பேசுங்க.
இங்க
நீங்க
வரல.
இதுல ஆன் எல்லாமே பேசலாம்.
Very good morning.
கேக்குது சார். நான் அவர் உள்ள வரல, வெயிட். நீங்க பேசுறது கேக்குது.
என்ன கேட்கல?
உங்களுக்கு கேட்காது. அவர் மற்றவங்க பேசுனதான் உங்களுக்கு கேட்கும். ஹாய்.
Very good morning to you, all of you. Bharat is an eminent resource person. Currently, he is an associate professor of Department of Earth Resources and Environmental Engineering, Hanyang University, Seoul, South Korea. He has acquired his PhD in Shivaji University, Kolkapur, India in 2012 for his contribution in biological treatment of industrial wastewater. He conducted his postdoctoral research during 2013 to 2014 in Hong Kong, the Baptist University, focusing on microbial sludge processing and wastewater treatment to upgrade the bioleaching method of sewage sludge. His research is focused on bioenergy generation with specialization in biofuels production, anaerobic digestion of waste organic resources. He is an eminent scholar. He has got rich experience in teaching. With a small introduction, I would like to invite Professor Mayur Kurade for this session. Welcome you, sir. Over to you, sir. Over to you, sir. Sir, excuse me, sir. Mayur Kurare, sir. Professor.
Dear professor, could you please unmute your audio because we couldn't hear anything from you. Professor, could you please unmute your audio? So, we couldn't hear anything.
Professor, kindly unmute, Professor. Your audio not audible. Professor, Karate Professor. Professor, audio is not audible, Professor. Not at all clear, Professor. You want- Audio is not at all coming, Professor.
Hello. Hello. Can you hear me now? Hello. Hello.
Yeah, yes, sir.
Now only audible.
Thank you. Thank you. I don't know what was the exact problem. I had to reach.
Kindly
Okay.
increase your volume, Professor.
Okay. Sure. Yeah.
Now it's clear, bro. Excellent. Audible.
Okay. Sure. Yeah. Sorry for the trouble. Okay. I will resume the presentation then.
You can proceed, Professor.
Okay. Sure.
You can proceed,
So
Professor.
okay. So I will start from the beginning and I will try to go a little bit faster then. So my name is Mayur.
Professor.
My name is Mayur Kurade. I'm currently working in Department of Earth Resources and Environmental Engineering in Hanyang University. So basically, we are working in environmental pollution research, and my present topic is to give you a glimpse of wastewater treatment technologies and organic waste reduction to bioenergy production. So the presently, before I begin, I would like to present some of the glimpse of my research lab. My research lab is led by Professor Byung Hoon John. So basically, we had a very large team of more than 25 researchers, and basically we are divided into two groups. One group is one group is conducting research in the chemistry, and another group is working in environmental biotechnology. So basically both these groups are the target is same, to resolve the environmental pollution problems. So in the chemistry group, some of the researchers are working in adsorption and photocatalysis to uptake the emerging contaminants from the wastewater. And presently, since last couple of years, we are also working on the CO2 reductions. As you may know that the CO2 problem is increasing, and more research is focused on this topic because of greenhouse gas emissions and carbon dioxide reduction. So our team is working on utilizing chemical and biological pathways to reduce carbon dioxide and convert it into useful compounds. uh, Regarding the environmental biotechnology research, we are utilizing different approaches, such as we are using microalgal biomass and plants to uptake the contaminants from the wastewater. As I have mentioned that we are using microalgal biomass, it can be also useful to convert it into utilize and convert it into useful biofuel products and value-added products. So we are also working on biofuel generation. And the lastly, in wastewater treatment, a lot of sludge is produced because wastewater is usually contained with the suspended particles, which are settled in the bottom. So we are utilizing this sludge to convert it into biomethane via anaerobic digestion. So before I go into the core topic, let me introduce you the typical wastewater treatment system process and exactly what happens and what we will do in this. So whenever the municipal and wastewater from the industries are coming into the wastewater treatment facility, it firstly treated with the primary clarifiers. Basically, here they use flocculants and coagulants to just coagulate the suspended particles in the sludge in the wastewater, and then the upper layer, which is a little bit clear, is transferred to the secondary clarifiers, where it is treated with activated sludge process, so that the dissolved matter is utilized and converted into the water and carbon dioxide. And then the secondary clarifiers, also some of the sludge is collected, and finally the upper supernatant is treated with UV and UV disinfection or chlorination treatment before it dispose off to the water streams. So the sludge collected here from the primary and secondary clarifier is transferred to this anaerobic digestion system, and then here it is further digested to minimize this sludge, and methane is generated in this process. So basically this first part is basically aerobic process, and the second part is basically the anaerobic process. So my research talk will summarize this both process in this presentation, and we will also see what are the other alternatives which we can use to treat this wastewater in the part one. And in the second part, we will check the how we can generate more methane utilizing this sludge biomass. So let's begin with the part first, that is aquatic pollution of emerging contaminants. So let me introduce what are the emerging contaminants here. So as you know that since the industrial revolution, we are producing a lot of synthetic chemicals for the better and standard of living. A recent report of Water Environment Federation reported that there are more than 80,000 different synthetic chemicals have been released into the into the environment in last 50 years or so. So I'm just focusing here on the emerging contaminants. So as the term suggests, emerging contaminants. Emerging doesn't mean that these are new chemicals. These chemicals are already existed in the environment. However, we did not know that what are the impacts of those chemicals on the human health or ecology. But a recent advancement in analytical chemistry, we are now able to identify these contaminants in very low concentration. We can even go up to ppb level to identify that what are the different chemicals are present in wastewater or any water samples. So there are different classes of these emerging contaminants, including pharmaceutical and personal care products for the related to human health. Then we are also having pesticide, insecticides, which are used for the agricultural purposes. And there are so on that flame retardants and synthetic fragrance, phthalate, bisphenol A, and there are many more. These contaminants are also categorized into emerging chemicals. So now what are the exact problem with which is related to these emerging contaminants? So let's look at this very interesting study which is conducted a decade ago by one of the scientists in USA. So in this study what they exactly did is, this is the Southern American map of American continent. So what they exactly did is they collected collected the river water samples from 9 different river basins, and at the same time they also collected the aquatic flora, mainly fishes, and they checked what is the contamination level and what is its impact on the fish. So interestingly, what they found is, 25% of the fish species were already having feminization issues, and 31% of these sites are heavily contaminated with these emerging contaminants. So this feminization usually occurs because of, whenever we take hormonal medicines such as estrogen, it can release from our body into the urine, and then it releases into the wastewater, and finally from the wastewater it ends up into the river basins. So the researchers have found that even our body cannot, intake all the 100% of this medicines into our body. It is not 100% absorbed into our body, so that it will finally end up as it is into the water stems, and then the aquatic flora will have very serious consequences because of this release of these contaminants. So this is a very, significant finding just 10 years ago, and you can imagine that in the country like USA, where that there is very strict regulation to control the water contamination, still there is, 31% of these sites are heavily contaminated with such pollutants, is really serious problem. Then we can just imagine that how about the normal countries or developing countries where there is not strict regulation which are followed to control this pollution. So let's talk about the available technologies. So basically we have physicochemical treatment technologies including adsorption, chemical oxidation, photocatalysis, and recently advanced oxidation process. These are very effective in some cases, but the problem is it may not be applicable to the, real field scenario, and it has some problem with the cost related issues. It also generates the secondary sludge. For example, if you talk about the adsorption or coagulation, it just separates the contaminants from the water phase, and then it will end up into the sludge. So again, we have to treat the sludge to get rid of these contaminants. So instead of that, bioremediation technology is more effective and economical. It is environmentally well accepted. And bioremediation technology, usually the chemicals are degraded rather than it is just removed from the wastewater. So basically we have a lot of different choices to use from bacteria, fungus, yeast, microalgae, and plants. So these are the biological treatment pathways. Bacterial and fungal studies are many more available in the literature. So we are trying to see whether we can use plants and algae. So first, let's talk about the microalgal utilization. The main advantage of using microalgae is because we can utilize this microalgal biomass after the remediation purpose for the production of high-end chemicals, including pharmaceutical and medicinally important chemicals. And we can also utilize it for biofuel production. So there are a lot of advantages of microalgae compared to the bacteria, as microalgal biomass is very useful. Besides, when we talk about the reactor setup, the microalgal reactor setup is much more easy. Why? Because bacteria require strictly sterile conditions, so that there will be a lot of contamination problems, and that's why there is a strict reactor conditions are required. Moreover, bacteria and fungal remediation technologies require a lot of nutrients for the growth of bacteria. Compared to these bacterial cultures, if you look at the microalgal cultures, microalgae just grow on very nominal nutrients which are based on the salts. That's why there is no contamination issues, and because of that there is no heavy reactor setup is required. It is also less energy consumption, and as I mentioned earlier, its high biomass productivity is always useful for the production of biofuels and other value-added chemicals. So let's look at some of the results what we have obtained. So basically we have tried to isolate a lot of microalgal cultures from the wastewater and other extreme environments. So basically we have found out that Chlamydomonas mexicana, Synodesmus obliquus, Chlamydomonas picimania and Chlorella vulgaris are much more effective in the uptake of these contaminants, and we have checked that these are very effective in the removal of some of the emerging contaminants such as carbamazepine, levofloxacin, sulfonamides, and so on. So we have, in this study, we have tried to use the removal of ciprofloxacin, which is one of the frequently used antibiotic in a lot of countries. So in the screening results, we have observed that Chlamydomonas mexicana had shown more uptake removal than the other microalgal species. The only problem is the removal is not that high compared to the bacteria. You can see here that mexicana shows only around like 13% of removal, where compared to bacteria it shows more than 50%. But I will let you know that what we can do to improve this removal efficiency later on. So first of all, we have to check the toxicity issues on this microalgal biomass, whether it has some toxicity on microalgal biomass. So at various contaminant concentration, we have checked the microalgal growth, and as you can see here that we have checked the growth from 0 to 11 days, and as the time is extended, there is increment in growth. However, you can see that at very high concentration of the ciprofloxacin, the microalgal biomass is inhibited. You can see that in the control, the growth is higher. However, in the higher concentration, the growth is more than 70% reduced. So high concentration definitely we can't use for microalgal biomass. But anyway, that in environment, if you see that higher concentration is like 40, 60 or 100 mg per liter, and in environment this concentration is never existed. In environment is less than 1 or 2 mg per liter. So that won't be a trouble. At very low concentrations such as 2 mg, in some cases that growth is slightly increased compared to the control, which is usually called as hormetic effect, which is basically low concentration enhances the growth of microalgae. But if you look at the specific growth rate graph here, you can see even in the higher concentration at later stage like 7 or 11 days, you can see that the growth of microalgae compared to the control is getting recovered. So what does that mean is microalgae, it also has some defense mechanisms against the toxic shocks given by these contaminants, and microalgae can fight back and still grow at a certain level. So as I mentioned to you earlier that the removal of these contaminants is very low in microalgal case. So just for example, you can see here that in control where we have added the 2 mg per liter of ciprofloxacin, you can see the removal is only 13%. So how you can increase it? By addition of some of the organic substrate as a redox mediator. So you can see here that by addition of nutrients or metallic micronutrients, we can increase this removal percentage. For example, without the sodium acetate addition in this study, the removal was only 13%. However, if we add the sodium acetate, we can increase this removal efficiency by almost 4 times. So in case of 4 mg addition of sodium acetate, we have identified that more than 56% of removal was obtained. It is basically because of the redox mediation reaction, which is served by this organic substrate. So what is the exact mechanism of this remediation? So basically in biotic approach, there is either bioabsorption, bioaccumulation or biodegradation. And in abiotic, we can see photodegradation and hydrolysis. So just suppose that this is the microalgal cells. So it usually the uptake removal is through the bioabsorption, where the chemical is attached into the cell surface, and then it is ingested into the microalgal cells, where it has the chance of either accumulation or further degradation by the enzymatic attack. So basically, there are a lot of enzymatic reactions are happening in the microalgal cells, including hydroxylation, carboxylation, and many more because of the presence of these enzymes, lacase, peroxidase, and hydroxyl radical attack. So before I move to further, I would like to just point out that once we have the remediation by this microalgae, we can recover the biomass and utilize for the biofuel purpose. So this is in this recent study. What we did is we extracted the lipids. We increased the extraction of lipids in the microalgal cells for the biodiesel production purpose. And what we found is because before the biodiesel production you must disintegrate the microalgal cells in order to extract the carbohydrate, proteins, and lipid present within the cells, and then you can utilize these macromolecules for the production of biofuels. So basically, when we use this microalgal biomass in fermentation by yeast, you can see that the carbohydrate is gradually decreased, and also proteins are gradually decreased. So carbohydrate fermentation usually gives you the bioethanol. You can see here that the bioethanol percentage is increasing, and at the same time the proteins are converted into higher alcohols. So higher alcohol will also increase. So basically, this graph shows that the dry cell weight of the yeast and the lipid increment in the yeast cell, because when yeast ferment the microalgal cells, it utilize the microalgal components and convert it into the highly rich lipid source. So once you have the higher lipids in this fermentation process, we can go for extraction using the ionic liquids. Compared to this conventional process, you can see that the ionic liquid extraction efficiency is very high. Within couple of hours, you can extract almost 100% of the lipids, compared to wait for 9 hours or 10 hours. And the most interesting fact of using this ionic liquids is once you extract the lipids from it, you just inject the carbon dioxide into this and recover the ionic liquids for its recyclable usability. So we did the recycle test here, and we identified that more than 6 cycles we can use this ionic liquids with the efficiency of more than 80% of lipid extraction. So in another study, we also find out that fermentation time of bioethanol production, we can reduce it by yeast fermentation, and we can also produce the higher alcohols using the protein biomass, and in the same process we can extract the lipids and convert it into the biodiesel. So basically, microalgal biomass usually contains 3 macromolecules. One is carbohydrate, second one is protein, and third one is the lipid. In most of the studies, they usually focus on only one component, either carbohydrate or lipids. However, in this particular study, in the same sequential process, we tried to utilize all these biomolecular components, including carbohydrate, protein, and lipids, to convert it into all forms of the biofuels. For example, in the first initial biomass, after the yeast fermentation, we converted it into the bioethanol. In the second fermentation process, using the another yeast species, we have converted proteins into the higher alcohols, and in the final stage, we have extracted the lipids and converted into the biodiesel production. So moving on to the next biological agent, that is plants. And if you use plants for the remediation purpose, then the process is called as phytoremediation. So there are different processes by which plant extract these chemicals from the water, by either phytostabilization, photo extraction from the roots. It can also to volatilize the compounds, and also transform these chemicals by its enzymatic reactions into the different tissue cells. So basically, since long time, plants are morely used for the removal of metals from the water or soil but there is not much attention has been given that whether plants can also uptake the organic contaminants and degrade it into the further chemicals. But when I was studying in India, my PhD advisor, Professor Govindwar from Shivaji University, has mainly emphasized and did a lot of research to prove that plants can also uptake the organic contaminants from the soil and water, and it further degrades it into the normal or common simple metabolites. So up to now, there are more than 350 species have been identified which can, uptake such contaminants and metabolize them into simpler forms. So let's look at some of the results here. So in this recent study, we used Ipomea aquatica, which is one of the aquatic plants, and we established this phytoreactor, a 30-liter reactor, and then we studied the removal of another pharmaceutical contaminant, that is sulfamethoxazole, and we checked the removal efficiency. So if you look at, plants show a little bit different trend than the microalgae, where the adsorption is going on at the first place and then it degrades further. So removal efficiency was very high, up to more than 80% for the lower concentration of the contaminants. However, if you use higher concentration, then the removal efficiency is little bit low. We also check the pigment concentration, whether these contaminants have some impact on these plants. But however, we didn't find any significant toxicity issues related to plants, unlike microalgae. You can also see this structural deformation. So this is the control tissue of root, stem, and leaf. However, after the treatment, there is not much, disintegration of those tissues. Hence, even at the higher concentration, plant shows a much more robust approach, and these are more tolerant to toxic shocks compared to bacteria or microalgae because of this rigid structure. So we did mass balance analysis here. So initially we infed the 1 mg per liter of the concentration of sulfamethoxazole, and this plant shows almost 72% of the removal. However, there are some abiotic processes, including photodegradation and pH change fluctuations, which might be involved in the degradation of these contaminants. However, it is very negligible, less than 1% observation. Adsorption was also observed on the roots of the plant tissues. However, it is also very less, 1%. As I said, the adsorption was obtaining in the first couple of hours, and then these roots ingest these chemicals inside the tissues. That's why in accumulation, you can see that there is 17% of this accumulation in the leaf tissues. However, the major contribution for the removal of this contaminant is the biodegradation, where more than 80% degradation or removal was obtained because of the biodegradation processes. So in the analytical characterization, you can see that there is only one peak of this chemical in the wastewater samples. However, there are some, plants usually generate a lot of metabolites. So there are, a lot of small peaks in the HPLC profile. However, when we add this sulfamethoxazole into the phytoreactor, you can see that there is a lot of generation of these small peaks, and there is a disappearance or reduction of this major peak here. So it suggests that this particular compound has been biotransformed into smaller molecular entities, which we later confirmed by the gas chromatography and mass spectrophotometry analysis. So you can see that this phytoreactor showed the degradation of this sulfamethoxazole into very simple monomers. And compared to the abiotic degradation, the phytoreactor shows very high degradation, high degradation efficiency to get the smaller compounds. So to finish with the conclusion of part one, microalgae and plants show very high tolerance toward the contaminant and pollutant exposure. Because of the time restrictions, I did not present all the data. We also did some enzymatic analysis, and in that we observed that superoxide dismutase and catalase enzymes in microalgae and plants are majorly involved in the protective mechanism against the external stresses. Microalgae plants are more efficient in removing pollutants even at high concentration, and it also involved in degradation of these contaminants into much more simple form. So moving on to the next part, which I will talk about how we can utilize this sludge and get more methane content from it. So for the bioenergy feedstock, we can utilize a lot of organic resources. Let me mention here that whatever is organic, we can utilize it to convert it into the biofuel. Whether it is crop residues, whether it is animal waste, whether it is food waste or municipal solid waste, we can convert anything organic into the useful biofuels. So the problem with the wood residues and crop waste is because wood residues and crops contain high amount of lignin, which is very hard to degrade, and that's why we have to spend a lot of process parameters and lot of investment into the pre-treatment of these residues if we want to utilize these residues for the biofuel generation. However, this all waste biomass doesn't require that much pre-treatment process because it doesn't contain lot of lignin residues in it. So that these are much more optional and interesting feedstock for the biofuel generation. So in this study, I would like to focus on fat soil grease. So basically, what is the fat soil grease? It's nothing but the a lot of food preparation where we used cooking oil, and because of we do not consume all the cooking oils, it goes into the water stream. And if you just dispose of your oil into the basin, this kind of problems can happen because oil is contains a lot of grease in it, and it can stick to the walls of these pipelines, severe pipelines, and then clogging, and a lot of severe problems will happen if we do that. And that's why the more recently the oil containers are provided to, or even in the basin there is a separate unit to just separate out this grease material. So we can utilize this fog, and we can either dispose it off using lot of these processes, either with municipal solid waste, with mix and screening to dispose of it into the landfills. We can also mix it with the dewatered solids. We can use incinerate it. We can treat it with the land treatment and direct application into the grease interceptor. We can also use this fat soil grease in dewatering. However, most of the these methods mentioned here doesn't provide you any benefits, because you have to add lot of treatment costs to get rid of this fog problem. But as this fog contains oil, we can use it for the biodiesel production, and as well as we can use anaerobic digestion process to convert this fog into the methane biogas. So both of this biodiesel production and anaerobic digestion seems very favorable options for us. However, for the today's presentation, I will focus on the anaerobic digestion. So what we did here, we incorporated 5% of this fog into the anaerobic digester, and we checked what is the methane generation potential of this. So this is graph shows you the daily methane production, and this provides you the cumulative production of the biogas. So as fog is very high strength organic substrate, it contains more than 90% of volatile solids. So it is very hard for the microbial population of the anaerobic digester to utilize and adapt to this situation. So you can see when we add fog into this, up to 40 days, we saw this inhibition phase. However, once the microbiota is adapted to this fog stress, it can convert this high strength carbon source into the very high methane potential. So once we add this fog into the digester, we can see that there is more than 200% increase in total methane generation. So it has very high potential for the biomethane generation. We did environmental microbiology to realize what are the different microbiota involved into the digestion of this fat, soil, and grease. And as the what we did is we simply checked the microbiota at different time slots at 0 time, 40 days, 80 days, and 120 days. So basically, this is the phylum level analysis, which usually shows that there is high percentage of bacteroidetes in the early phase. However, when the latter phase, when the biomethane production is very high, you can see that firmicutes and bacteroidetes were increased tremendously at the latter phase. This particular heat map indicates the major genera level organisms in the digester, and as you can see that Syntrophomonas, Clostridium, Methanosarcina, and Methanoseta were the major contributor into these genera. So basically, these Syntrophomonas and Clostridium species are usually responsible for the degradation of oils, or it is responsible for fatty acid oxidation. That's why the percentage of these particular microorganisms increased into this into this reactor. This graph shows you that metagenome comparison using the Venn diagram, where there are common operational taxonomical units can be observed. So to simply give you a glimpse, you can see that this is the central diagram where 366 common operational taxonomical units were found in all those 4 samples. However, at each time point there is different diversity of microorganisms, and that's why whenever you add different nature of the substrate into that, the microbial population will change its nature. This principal component analysis also reflect the similar things. You can see that when 0 time sample, to just interpret this figure, if these points are lying away from each other, it means these samples are very diverse from each other. So the microbiota present in this control sample was too much different than the 40-day sample. However, 80 day and 120 days sample points are lying a little bit close to each other. It means that the microbial diversity in those 2 samples were pretty much similar. The most important factor here I would like to present, in the 0 time, when we accumulated the methane generating bacteria together, at the beginning of the time, 0 time, Methanoseta was dominating into the reactor. However, after the addition of fog, you can see that Methanoseta was changed to Methanosarcina. So Methanosarcina population was overtook by Methanoseta, and the major reason is because Methanoseta has different growth kinetics than Methanosarcina. And the thing is, when oil are degraded into the simple monomers, the final product is acetate. And because we added a lot of oil into the digester, acetate concentration was very high in the reactor. You can see that the initial the maximum concentration this Methanoseta can tolerate is less than 3000 ppm. However, Methanosarcina can tolerate around 15,000 ppm, which is 5 times higher than the Methanoseta. And that's why the methane generation in this process was majorly led by Methanosarcina species. So conclusion of part 2, I would like to point out that addition of fat soil grease inhibited the methane production for certain period of time. However, it increased very high methane content, which is more than 200%. During the digestion of fog, there are major microbial groups involved in this digestion process, including acidobacteria, bacteroidetes, urea quota, firmicutes, and synergistes. And as the time proceeds, there is very distinct microbial population with varying dominance of each group. And the main importance of this study is we check that Methanosarcina was the main methane generating archaebacteria, which was responsible for digestion of this fog and converted into the methane. So these are some of the research publication from our group, which we published in Chemical Engineering Journal, Bioresource Tech, Trends in Biotechnology, and so on. Even water research. Some of the book chapters. We have one patent on this biomethane study, which I presented here, which we also recently technology transferred to one of the company. As you can see in this photo, we have very big research group working with Professor Bang Hun John, and he really appreciate and respect the Indian staff. So you can see more than half of the staff is filled with Indian staff. You may know Dr. Prabhu, which is a close friend of some of you who recently joined VIT. He was also working with us more recently. So I would really like to thank especially Dr. Rayapan for giving me this opportunity to present my work here, and also I would like to thank Dr. Prabhu for introducing me to your conference. So I guess that I have presented work in like very fast way. Sorry about that because of the initial technical glitch in the microphone. So if you have any questions, I would really like to get feedback.
Thank you, Professor. Hello.
Yeah.
Thank you, Professor. Very much appreciated and excellent presentation, Professor. We came to know that you are talking microalgae from biodiesel and sludge to biodiesel. There are two category you have given lecture. It was very much appreciated. It was a very informative, Professor. Behalf of our management, Arul Anandar College, and behalf of EFC 2023 team, we are grateful and honor you, Professor. Thank you very much for your lecture. And participant, you have any queries or questions, you can ask now.
Dear Professor, could you please show the instrument that is available in our laboratory, because we could see some of the instruments. If we could see, our students will be get benefited out of it. Could you please?
Sure. Actually, right now I'm. If you just allow me, I can show you some of the major instruments. Currently, actually, I'm presently I'm giving this presentation from the molecular biology setup. So the instrument you can see here is the next generation sequencer, which we used for the microbial population identification in a lot of environmental samples. So this instrument is also attached to this, which is library preparation for this next generation sequencing. We also have the molecular biology setup, including RT-PCR and BID preparation. So this is all setup you can see here is usually for molecular biology setup. And we also have a couple of different labs for working for chemistry and biology. So Hanyang University has very high instrumentation facility. The mostly analytical instrumentation, we can go to the central facility where you can do imaging techniques, XPS, FTIR, XRD, and a lot of XPS, a lot of other stuff is available in Hanyang University. So if you have any help, we are very happy to help you.
If there is no question, over to the desk.
Sorry?
Professor, thank you very much for your lecture. It is very informative and useful. Our students will get to know through email. If any queries,
sure.
We'll
No problem.
Okay.
Yeah, I would be happy to help you. Yeah, thank you. Thank you for this
opportunity. Thank you very much, Professor.
Thank you.
Okay, thank you very much. Over to
Professor, thank you very much. As head of the Department of Chemistry, my professor, thank you very much. Future, if it is any queries or anything, we will contact over the mail, Professor.
Thank you.
Thank you very much for your acceptance and you are highly informative, resourceful, informative lecture.
In fact, I was a little bit worried because when Dr. Prabhu told me that this is the conference, and when I saw the program, most of the candidates are from the chemistry faculty. So I was a little bit worried whether I will be a good fit on this conference. But Anyway,
you are a completely resource person, Professor. Happy, very happy. And a
thank you,
novel
Dr. Raipan.
topic also.
Thank
Novel
you, Dr.
topic,
Raipan. Thank you.
reachable.
Yeah, thank you. Thank you for giving me
Thank
this
you very much.
opportunity.
You are all the thing.
Oh, thank you, Professor. Thank you.
Thank you, Professor. Thank you. Thank you. You can leave, Professor. Next
Okay.
Professor will start the
Okay. Thank you.
lecture,
Thanks a lot.
Professor. Thank you,
Okay.
Professor.
Bye. Bye.
Mahendra sir, shall we start next session, sir?
Yes, sir. Yes, sir.
Yes.
Yeah.
So I am very happy as a head of the department. I am sharing this session also. So first of all, I wholeheartedly welcome this session. First of all, I thank Dr. Mahendran Velachami. So he is a person I have gone through this profile. It covers 8 pages. Highly rich profile. So he is a art person. He is a well renowned and latest topic. He is also a regarding his education qualification. Sir, he has completed his undergraduate in the American College and postgraduate National Institute of Technology, Tiruchirappalli, MSc Applied Physics. Then he moves to Indira Gandhi Center for Atomic Research, ICAR. That is for his PhD work. Sir, so far, rich experience, starting from after completing his PhD degree, 2015, from his postdoctoral research, physics of complex fluid and next associate soft matter laboratory Slovenia under the Professor Dr. Frieder Mukeli, and also he is also a teaching experience and the research area interest micro droplet laser and cholesteric liquid crystal photonics, and another one area colloidal forces stimuli responsible nanofluids for the thermal transport, development of advanced functional materials and sensors, and ion absorption or at solid waste, solid water oil interfaces, enhanced oil recovery, and also synthesis and characterization of nanomaterials. He is also experimental expert from nanoscribe photonic professionals and microfluids, magnetic changing technique. So these are the fiber optic sensors and quartz crystal microbalance. These are the latest experimental expert. And software, other latest software also, he well versed in the software. Sir, his achievement, PhD during his PhD tenure, student award, PhD student award 2014, and also research fellowship at Department of Atomic Energy, and 2010 MSc project award, 2009 award of state eligibility for lectureship. Research highlights in India, so many things, and a reviewer of Elsewhere reviewer and Springer reviewer, American scientific reviewer. These are the reviewers. To his credit, a list of peer-reviewed publications. Sir has published so far 22 papers, national, international reviewed journal with high impact factors. A list of conferences, so many he has attended international conferences. To his credit, highly resourceful resource person with us. Thank you very much, Professor, for accepting this international conference. Since we are conducting from 2001 onwards, this expanding frontiers in chemistry. So far, we have received 80 research abstracts, both oral and poster presentation. 4 abstracts from the abroad, Malaysia, South Korea, and Singapore, and other countries also. Thank you. Over to Dr. Mahendran, sir. Thank you, sir. Now over to sir.
Thank you, Professor Rayappan, for this introduction, and also I take this opportunity to thank the organizers for giving me this opportunity to share some of my line of work. And it's really worth mentioning that the efforts you have put in organizing such an interesting line of talks for the benefit of students, and it's highly appreciable. And yeah, so to the audience, so you have been listening to different line of talks more related to chemistry and biology, and this talk will be little bit branched out in the sense, so this talk I will try to present a physicist perspective of where are the other horizons where chemistry can help in improving or taking more technological problems forward. So the theme of the talk is probing molecular interactions at interfaces. So today I will highlight 2 interfaces. One is solid liquid interface and the liquid liquid interface, and I'll also show how we use this basic understanding to solve some real-time industrial problems. So when we talk about molecular interactions are very fundamental interactions which varies from short range to very long range, and these forces are integral part of many products which we encounter in day-to-day life. Here I list some interesting applications. As you can see here, the left 3 essentially deals with the short range interactions, the application mainly based on short range interactions, and the right 3 applications where you can see the macroscopic applications, like the application goes in meters, kilometers, and so on. And also at the boundary we have this is one where the interface of nanoscale and microscale understanding is essential to make mu functional materials. Since the topic is vast, I don't want to explain all the concepts, but I'll try to explain in a simple way, with 1 examples from liquid liquid interface and 1 example from solid liquid interface, and then I'll show how we address the challenges in the real time. Okay. So let me start with the liquid liquid interface. So this part I divide into 3 parts. So I'll introduce the system, that is our material which we use, and then methods, and then I'll show some test cases, then I'll show some applications. So the system here is emulsion. Emulsion is basically, it is a biphasic system where we have 2 immiscible phases, for example oil and water. When we mix these such immiscible phases, what happens is they break into droplets, and then over a time they phase separate. This is because of the high interfacial tension, and also this system the immiscible systems are thermodynamically unstable. That means the order of kBt energy is enough to phase separate the systems. So to avoid this phase separation and to make this system more kinetically stable, usually we add some stabilizers like surfactants, organic molecules, or even particle can do this thing. So when we break this biphasic system in the presence of stabilizers, we end up in colloidal dispersion, which we call it as emulsion, where the less volume phase constitute a dispersed phase and the large volume phase constitutes a continuous phase. Based on the continuous phase and the dispersed phase, the emulsions can be categorized into oil in water or water in oil, or even we can have multiple phases in a single continuous phase, like oil in water or water in oil. So this kind of system is called emulsion, and these systems are not new to us because we are using such emulsion products in our day-to-day life, starting from the morning. For example, here is one interesting example. Milk is the nice example for oil and water emulsion, where the fat globules are stabilized by the milk protein called casein, and also we have some calcium ions which stabilizes these droplets against flocculation. And if you go to the other extreme, where again this example from milk, so when we ferment the milk and we take butter, the constituent of butter is just opposite to milk case. In milk, water is the major constituent, and in butter, the water is less constituent with water and more in fat. And the other lotions which we use are such an example of multiple emulsions. So the emulsion systems are integral part of our day-to-day life. And also, other than this, we have different food emulsions, paints, salad, and medical medicines, and in different forms. So today's topic, I will highlight more on water in oil emulsion. Sorry, oil in water emulsion. Here, the dispersed phase is oil phase, and this oil is not normal oil, and we have a special oil called ferrofluid oil. So this ferrofluid oil consists of nanoparticles, which are iron oxide nanoparticles, which are sterically stabilized by surfactant against flocculation. And the typical size of these nanoparticles are 10 to 12 nanometers. And then these nanoparticles are super parametric. That means when we apply magnetic field, the particle behaves like a tiny magnet. Nanomagnets, when we remove the field, it behaves like a normal particle. And that means the particle doesn't remember what is the magnetic field it experiences, right? And then because of this super parametric nature and the finalization, what we have is we have a single phase. When we disperse this particle in any oil, the particle and the oil coexist as a single phase. The movie here shows that when we apply magnetic field to this oil, what you can see, the whole liquid is respond to magnetic field as a single entity. This is the beauty of this oil. So by applying magnetic field, we can manipulate the flow of this ferrofluid oil, and you can clearly see there is no phase separation. There is no particle present at the bottom, right? And this is one of the interesting thing with this ferrofluid oil. So it's a single phase. Now, for the preparation of these emulsions, what we do is we go with the very low volume percent, like 5-8% of this ferrofluid oil, and then more than 90% is only water. So we shear this oil in the presence of water with some stabilizers. Then we end up in breaking the layer of oil into small droplets, and these droplets are different in sizes. Then we use technique called fractionation. It is like centrifuging and then separating the large droplets by sedimentation, and then separating the smaller droplets which are at the top. And then we do this process multiple times to obtain more or less uniform droplet size of 100 nanometer, 100 to 200 nanometer. And this is the typical cryo frozen TEM image of these droplets. So size is like 100 nanometer. And then during this method, another advantage is we can choose different stabilizers, from single molecule surfactant to large molecule polymers. So The freedom here is by choosing the stabilizer at the synthesized phase, we can have droplets with ion stabilized or droplet with macromolecular interfaces. And as you can see, the size varies from 100 to 200 nanometer. It basically depends on what kind of stabilizer which goes at the interface. And these stabilizers can be result in different fundamental interactions like electrostatic, steric, or electrosteric. I'll address all this in the coming slides. And another point here to note that the droplet volume fraction is very, very low. That means we have less than 1.1 volume percent. That means the volume of emulsions is more than 90% is water, and we have very small amount of this magnetic nano fluid in the system. And what is interesting about this system is when you take this sample and put it in a slide, and if you observe under microscope, what you see is this Brownian motion and behavior of any colloid. And the droplet cannot come together because of the interfacial stabilization, and the stabilization provides kind of repulsive force that prevents the droplets to come closer. Okay. And this is normal, so it is not surprising. When we apply a small magnetic field, very small magnetic field, what you see here is the droplets try to form one-dimensional lattice. And if we see this lattice structure under any white light illumination, even in the room light condition, what you see here is you see different colors. The sample appears in different colors. This is because the lattice spacing which is formed under the magnetic field is in the order of visible wavelength. So the lattice spacing can be controlled with magnetic field. As you increase, this is at zero magnetic field, and as you increase the magnetic field, the droplet spacing starts decreasing, or the lattice spacing decreasing. Then it diffracts colors from red to violet. And this tuning of lattice spacing is purely by external field. So here we apply magnetic field, which is non-contact, and then you can tune the lattice spacing externally from visible, the entire visible range, from UV to red, and even we can go to IR if we needed that interaction. And once the field is removed, it is completely coming back to normal. So we can reuse this formation and reformation multiple times without compromising anything with the material property itself. So after looking at the color change by the magnetic field, we thought of testing this as optical sensor for different analyte detection. For this, what we do is we keep the lattice spacing fixed by using known magnetic field. Now then material appears in one color. Then what we do is we just add some known species or unknown concentration of species into the system. At the same magnetic field, the lattice spacing, change in lattice spacing is only due to the additional species. And if you see here, for example, this case, we added potassium. So when we include potassium, what we see is the lattice spacing starts decreasing over the concentration, and then we have clearly a linear dependence on the concentration and the shift in the color, right? And also not only the blue shift, we can also have red shift. As you can see here, this is the case where the same fixed magnetic field, when we introduce glucose, you see it is the peak shifts more towards red. And we have very good linear regime. And this is a simple idea. This opens up to design a more functional and ion specific or species specific optical sensors. So this system, as I mentioned earlier, this system is very user friendly, and you can manipulate the interface during the synthesis process itself. And if you tag the interface with more specific taggers, then we can have more specific, selective, and easy to read optical output based sensors. And also this another interesting application based on this response, which is kind of in a simple way I can put it like this, is a magnetic field sensor. As I mentioned earlier, these droplets respond to magnetic field, and it forms a lattice spacing. Right. When we have uniform magnetic field, the entire volume of sample experience same magnetic field, and then the droplet lattice spacing is more uniform. What happens if we have non-uniform magnetic field? For example, in this case, the magnetic field variation looks like this. It's like you see the pattern, this pattern, right? When we have non-uniform magnetic field, how this responds? So when we have non-uniform magnetic field, the emulsion undergoes different orders. This order represent the field pattern. So from the field pattern, we can also image more complex geometries like this. So if you have a geometry, a defect geometry like this, and then when we magnetize this material, this kind of material, wherever there is a defect, the defect shape induces the leakage of magnetic field on the other surfaces, something like this. So when there is a defect, we create non-uniform magnetic field, and this magnetic field can be mapped with this emulsion product. And then what you see here is you can see the shape of the defect which causes this magnetic field. And here are some more interesting examples, and this is one way to just to show demonstrate how we can use this material to present the leakage fields or non-uniform magnetic fields. So if one can make magnetic field in the form of picture or in the form of some secret code. So this material, we can store the information without magnetic field, and when you apply magnetic field, then only you can read what is the message hidden. And this is the proof of concept idea for this purpose. Okay. So, but our main interest was to address the colloidal interaction, right? So colloidal interactions are so the main interest was to study the interactions at the interfaces, and this idea came up from an industrial partner there, where they had an industrial formula, in which they had problem of phase separation in a short span of time. So if you think of colloidal products, usually these are, as I mentioned earlier, this can be single emulsion or double emulsion or multiple emulsions. Basically, mixing of two or more immiscible fluids, then with some surfactants or with one or two stabilizers. In the industrial formulation, we have multiple such stabilizers present, multiple miscible phases present in the same product. So their understanding of this complex interface is very important, and it's also interesting, challenging problem for us. And also, this understanding can be used to understand the artificial knee joints, where we are using artificial bioproteins like liposomes, hyalurons, in combination with different ionic concentrations that stretches or collapses these polymers or proteins. And when we have such collapsed or extended structures at two interfaces, and these two extended layer gives a kind of lubrication for the knee joints. So this is the basic idea of the artificial knee joints. Okay, so the basic line of question here is, how do we choose appropriate polymer or appropriate ion concentrations or appropriate stabilizers for different applications? So here is for more industrial application, here is more biological application. So how do we use this? So the simple answer is to understand what kind of interaction is happening at the systems. So here we are going to use this emulsion, whatever we have shown before, as a model system to study such interactions in a simple system and more complicated environment. And how do we measure this force? we measure the force by using this technique called magnetic chaining technique. This is customized. This is not a single instrument. So this is combination of different instrument which we assemble and to for the specific applications. So the main integral part of this technique is electromagnet. This is solenoid coil which gives field along the center, and we create magnetic field by applying the current. So by applying precise current value, we can control what is the magnetic field produced by this solenoid. And then we use white light source to illuminate the sample, and then we collect the backscattered light through a spectrometer, and then we analyze this. So what we get out of from our experiment is we get the reflectance spectra, that is the diffraction peak, as a function of wavelength, right? And then we convert this spectrum into force versus distance curve. Here what I show is log scale, and as you can see here, with this technique we can measure piconewton force. Such a tiny and weak forces can be precisely measured in the range of less than 100 nanometers. So this is the freedom which this technique allows you. So you can measure even weaker interactions which are in the range of piconewton, and we can probe down to sub 10 nanometers. So the basic idea of the interfacial distance is from the Bragg's law. The Bragg's law tells that the interdroplet spacing or the lattice spacing is proportional to the diffracted wavelength, right? So we know the wavelength, and from there we get this d, and d is represented as in the form of interfacial distance. And then this force, how do we get this force? So what happens? So if you want to understand how we calculate this force, we have to understand what happens to these droplets or this emulsion system under magnetic field. When we apply magnetic field, the system starts from kinetic, that is from the Brownian motion kinetic state, to mechanical equilibrium state. So without magnetic field, you see the droplets are in kinetic motion because of thermal energy. And then when we apply magnetic field, there is a competition between the thermal energy and something else. What is this something else? This something else is from the magnetic contribution. So as you know, the droplets are made up of ferrofluid oil, and these oil constitute tiny magnetic nanoparticles. And these nanoparticles are super paramagnetic. That means when you apply magnetic field, then they remember the magnetic field which they are experiencing. When the field is removed, they behave like a normal particle. So you can see all kinetics of thermal motion, right? So when we apply the magnetic field, what happens is there is this competition between these two forces, and then over a time, over the critical magnetic field, the droplet, the induced magnetic field in the droplet overcomes the thermal energy, and then they are more static, and then there comes the balancing equation. So this balancing equation is the balancing of forces, the repulsive forces, which is mainly due to the stabilizers at the interface, and then attractive forces are due to the magnetic field which we apply to the system. So once we balance these two forces, that we create a lattice of droplets in a liquid, in water. So we can create lattice of droplets in water, and then we can tune these interfaces by the magnetic field. So to make it simple, the attractive force is mainly depends on the particle size, the magnetic nanoparticle size, and their magnetic moment, right? So we can fix this force because this is experimentally measurable quantity, and this magnetic field is what we apply from solenoid. So we know these two numbers, and then that means in the force balance equation, we know what is the attractive term due to magnetic field, right? So this is one side we know. And then when we go to, as I mentioned, this technique allow us to go below 10 nanometers. And when we go below 10 nanometers, we have to worry about van der Waals forces, because in short ranges, the van der Waals forces are very strong. So you can see here. So when we go down to 10 nanometer, below 10 nanometer, the van der Waals forces are very significant. So we have to take into account of this van der Waals forces. Again, these are a material property. This can be for given material composition, we can get these numbers, and this is precisely calculated, and this is well reported, nothing magic here. So this is again a feed from our end. Then what we have is, now we have the attractive term, attractive force, that is balancing the thermal energy plus the repulsive forces due to the stabilization. Now our interest is to address what are the cause for these repulsive forces, right? These are, may we know, these are from the stabilizers, and we can address these different forces by the balancing equation, and I'll show such examples. So let's go with the very basic system where we have a droplet with particles, magnetic nanoparticles, and the interface here is negatively stabilized. That means we have a surfactant, which is sodium dodecyl sulfate, which gives us negative surface group at the interface. And then when we have such system in a electrolyte, essentially we end up in double layers. The idea is, can we measure such a double layer interactions, and can we quantify what is the charge at the interface? Can we quantify the ion concentration, the electrolyte concentration in the bulk? Yes, we can do that. And this is one example where we measure the electrostatic interaction with different electrolyte concentration. So what you see here is, in a pure solvent condition or with zero electrolyte, the red curve, red open data and the line, this is the data. This shows that the force decays exponentially over the distance, and the decays at very long distance at 50 nanometer and so on. And this is the typical characteristics of double layer electrostatic repulsion. And then this kind of repulsion can be modeled with the DLVO theory, which takes into account of the surface charges and this kappa, which talks about the electrolyte concentrations, right? But what happens when we include more ions into the bulk? We are introducing more electrolytes, and these electrolytes essentially go to the interface and they try to screen the actual charge which is at the interface. So in this case, the actual charge is a negative charge when we put more cations into the system. So these cations try to screen the surfaces over some concentrations in the bulk. If you observe from the bulk, we don't see what is the charge, what is the original charge at the interface. We see more the screen charge, and if we increase even further, we see like hard sphere repulsion, where the charges are completely neutralized, and then this like two hard metal balls or two hard solid balls, when we bring them closer, the force is exponential. This is what you see here. So with this technique, we can also not only see this electrostatic interaction, and also we can see even hard sphere interactions. This is very interesting to or worth mentioning, this technique. And then from this force data, what we get three interesting parameters. One is the first interaction length, which we define as the starting point of the force. This is where the balancing of these two forces starts, right? When the kinetic motion is arrested, and when the system attain mechanical equilibrium, then they feel the presence of the other droplets. This is the starting point of the interactions. And then we have decay length, or the kappa, which gives this electrolyte concentration in the bulk, and the magnitude of force, which tells that how fast or how slow the screening is happening over the time. And this is the comparison of the quantities which we measured from our technique and the theory. So as you can see, this method can predict or can measure the salt electrolyte concentration with more than 90% accuracy. And this is the advantage or the performance of the system. Okay, then let me go to the other aspect where we have interface. Now the interface is no more charge. Now I put a macromolecule which are amphiphilic. So amphiphilic, so the molecule which is having hydrophobic and hydrophilic part in random distribution. If you put such a molecule as a stabilizer, this hydrophobic part will prefer to go into the water phase and into oil phase, and the hydrophilic part go into water phase, and this gives kind of sterically stabilized system. So steric stabilized system is nothing but the stabilization is mainly because of the conformation entropy of this macromolecule. So you put a macromolecule and they undergo different conformations, and this conformation provides stability against flocculation, right? And here, yes, this is the case. Now it is more interesting to observe. Here, as you can see, the force is very clear. It follows exponential over distance, but the decay is not following electrostatic double ray repulsion. Then what it follows? It follows exponential, and the decay scales with lambda. This lambda is in analogy with radius of gyration of polymer. For example, if I have the interface, if I have polymer at the interface, and this polymer in a good solvent condition, the natural maximum free volume of any polymer is assumed to be a hypothetical sphere, and the radius of the sphere is called radius of gyration, and this is one parameter which characterize polymers in a solution, in any solution. Okay, so in this case, in a pure solvent condition, the force decays exponentially with the radius of gyration of the free polymer. And then if we change the solvent condition from good to bad solvent, we know that the polymer prefers to collapse or more conformation or collapsed conformation, right? So we tested this. So here what we did is we added small amount of 50 ppb of electrolyte. Already we are changing the solvent condition from good to bad, and you can see the force profile changes, and the first interaction or where the droplets start feeling the other droplet is already changed by 10 nanometer, almost 10 nanometer, right? That means already there is something happening, and if we keep on increasing this concentration, you will see the interaction goes more towards a shorter interfacial distance, and this is one indication that there is something happening to the extended polymer. And from the force profile, what we can say that yes, yeah, we see this is collapsing here and this is extended here. And what is the proof? The proof is this number, this lambda. So if you look at this lambda, as you can see here, with the zero electrolyte condition, we have 9.5 nanometer, which is the radius of gyration of polymer used in this case. And as you change the concentration of electrolyte, you can see it undergoes the lambda collapses. That means the dynamics of these polymers at the interface is more like conformational or collapsed one. Okay, and this is one method which is applied in artificial knee joints, of course. Not only this simple interaction, we can also see very feeble effects like this case. So here what we want to address is we want to address the ion hydration. Ion hydration is one of the I think in my perspective, it is one of the least explored topic in the forces, and only few groups are working on ion hydrations and then effects on layer absorption and so on. And we also address such a problem. So here we take 3 test cases where we have 3 different ion sizes. So when depending upon ion sizes, the hydration or the tightly bound water molecule around the ion changes. When we have smaller ion, there are more tightly bound water molecules, and as the size, the ionic size goes higher, you have less number of water molecule, and these are loosely bound. So can we distinguish such a tiny thing happening at the few Armstrong level? Yes, we can do that also. So here the system is, so we put polymer stabilized interface, and then we study the effect of collapse, how this polymer collapse by different hydration cells. So you can see here, this is monovalent ion which has weakly bound or less hydrated, and iron Fe3 3 plus, which is strongly hydrated. And you can see here the weakly hydrated has very less effect on collapse, but at the same time, when we have this strongly hydrated um, ions, collapses faster. You can see here up to 5 millimolar sodium ion has very little effect, but when we have 0.1 millimolar of 3 plus, you can see the effect is already visible, right? So from this force, indirectly we can measure, we can characterize how strong or how weak the ion hydration is, and these informations are very useful, as I mentioned earlier, for the artificial knee joints. So which ion to choose, and which polymer to use, and what is the appropriate concentration for confirmation, and how to recycle this a number of times. And this is the basic feed for such application. And the last case, which is the motivation, is from the industrial partner, and they had a formulation which was having problem in terms of stability, or in terms of the expiry date. So the product sustained very short time, not for long time, so they want to improve the expiry date of the product. And then the composition I cannot reveal, but we have a model system. The model system here is, okay, we have one electrostatically stabilized system where the interface is stabilized by charges, and we have one system which is stabilized by polymer. And then we introduce milk protein. This is nothing but milk protein, which we separated from conventional milk, since the industrial formulations are patented, so we cannot share it actually, but this is the typical system. And then and then what happens is, we have two systems. One is system stabilized by electrostatically, and in this case, when we add the protein into the system, not much change. Not much change is observed. Still, the product is okay. When we go to this electrostatically stabilized, when we go to this polymer stabilized system, where the interface is with the polymer, which is a neutral polymer. That means there is no charge at the interface. The interaction is mainly due to as I mentioned earlier, the interaction mainly due to the conformation entropy. If we introduce this formulation, the protein formulation, into such system, something strange happens. So you can see this is one signature, that the polymer at the interface undergoes crazy collapse over the additive, and this is what we see here. It changes a lot. It's significant. And then we also did some additional measurements to measure how the surface charge is changing over the different concentration of this additional formula into the product. And then to our surprise, we ended up something like this. So you can see here, this green dot, which is the surface charge. The surface charge gets more negative. We started from nearly zero charge. This means that interface is only with the start formulation, that is with the polymer. And when we include this formula casing into the system, as you can see here, with low concentration already, the interfaces become more negative. That means the polymer cannot be charged, because this is a single molecule. The casein itself a single molecule which has charges, which is a protein with some phosphate groups. So this we know that this charge, negative charge, could only come from the protein part, not from the polymer. And And then at the same time, if you see the size of the droplet, it starts decreasing. That means something crazy happens at the interface. And we did further investigation, and then what we understood is when we start this the collateral formulation, this is the case. And when we introduce a small amount of protein into the system, what happens is there is a competition for the interfacial site or the surface site. So there is a competition between this polymer and this protein. So at low concentration, they try to coexist. And when we go to high concentrations, this interface is completely removed. Right, whatever the original stabilizer is completely removed by this protein, and then now we have a stable system, but the system is not the original system. Right, then this is the understanding we got. And then this is one way to understand what is the challenge the industry faces. And then they had to reformulate the starting point when they want to use this protein together with the formula. Then they have to reformulate the interface, and then we have proposed other formulations to start with, and then we had successful stabilized product at the end. Okay, so to briefly summarize this part, so this technique is user friendly, and it offers more of freedom to choose different interactions, and it can address from fundamental problem to real-time industrial problems. Like, if you want to understand fundamental interactions, short-range interactions or long-range interactions, and also to address a real-time problem, what we see in the market can be addressed by this technique. And Professor, how much time I have? Sorry, I can wind up accordingly. Hello, Professor Ayapan? Okay, so I'll stick with my time. So the second part, I would like to talk about the solid liquid interfaces, and then how do we use understanding from these interfaces for again another interesting industrial problem. It is from our
Professor, you can
yes.
take 5 more minutes, Professor.
Okay, then I will wind up quickly. So I'll show interesting thing, something. Okay, so the problem was this. This is from the industrial partner. The problem was this. The problem is they have oil, this crude oil in oil reservoirs, and these oils are very sticky to the mineral pores. And they want to understand why it is happening and how we can improve the wetting at the interface. This is a model system. What you see here is you see oil at the surrounding, and then we have water at the interface, and this is a model clay. And as you can see here, this is interesting to watch. Yes, so what you see here, you see, yes, it goes, and then over a time, the surface become more oil wetting, and we want to understand this. And the system is more complicated. This has oil, this has ions, and it has some macromolecules. And then we addressed all these problems by techniques like this. For example, when we use this ellipsometry technique, what we get is the refractive index change over the over the bulk. And the freedom here is we can choose even sub nanometer thickness from the interface. If you want to measure single ion layer absorption, yes, it is possible with this technique. So you choose your absorbed layer thickness, and then we get this ion, that is change in refractive index. From the refractive index, we can quantify how many number of ions absorbed at the interface. So we can have single layer, multi-layer, and then to the bulk. We can have this freedom, and this is what we get. We get to know the number of ions absorbed per unit nanometer square. such information we can get. And then we use another technique to study the absorption of the model organic. And as you can see here, so here we quantify how much of this large organic molecule absorb over unit area. And then we use these two informations to explain what is happening in their industrial problem. So the problem is here they have some organic and some ions and then oil and the mineral. So what happens is, this is what happens. There is a synergy between these two, ion and this organic molecule. So these ions and organic molecules, they come under synergy and they make this bridging, and this bridging is the major culprit for this sticking of oil to the mineral. And then now now we need the chemistry to break this interesting bond. And then we formulated 14 compounds and compositions to break this, and all the 14 were successful, and it is tested. It is running real time in British Petroleum oil reservoirs. Yeah. So with this, I summarize this part. And yes, so we can use ellipsometry and quartz microbalance to study molecular absorption and also ion absorption of the interfaces. And we can take this fundamental learning to real-time macroscopic applications, like in this case we are using this understanding in oil reservoirs. Yeah, this is a $1 billion industry, so we got a lot of money, of course. So yeah, that with that I would like to conclude. And my goal is to understand my interest is to understand these interactions at the interfaces, and then bring it to real time and more industrial problems. And yes, and I would like to acknowledge my collaborators from these institutes, and also all your kind attention. Thank you.
Professor, thank you very much. Now the question, audience question. Audience, you can text the question or you can ask the any audience question. Professor, I'll brief your talk, invited talk, Professor. That is, you are started with the molecular interaction of interfaces, starting with the MSS, super paramagnetic nature. I think through your talk and water talk interaction, it is happening. But it is helpful for the society, helpful for the cities. I have some question, Professor, regarding the droplet induced magnet. How long it maintained its behavior or maintained its magnetic behavior, Professor?
Okay, so these particles are super paramagnetic, and that means they don't what do you call? They don't retain the field. The moment you apply field, it behaves like a magnet. The moment you remove, it will become normal material. So there is no coercivity of the material. So this has extremely long lifetime. So only problem is if you use electromagnet to apply electric magnetic field, the electromagnetic generates heat. Then heat causes problem for the magnetization. That is the only problem. So when we have a heat generation around the system, then we have problem of low magnetic or loss of magnetization of these particles. Otherwise, if you use a permanent magnet, then
Yeah.
this magnetization is last forever, I would say.
Oh, nice, Professor. That is regarding the separation. It is in the kind of interaction study. It helps to separation in the, especially in the industry. What about
Yeah.
the biological system? What ways in a medicinal field, the interaction also in medicinal values. Is there
Yes.
any
Yes.
studies going on biological interaction in the nanoparticle range?
Yes. So currently, my previous group, they are looking at how to use these nanoparticles to cure, to locally kill the tumor tissues by temperature. So they apply RF frequency, and then these particles generate heat locally, and then we kill the tissues locally. So how we to go to the specific target, we functionalize the interface with the target proteins, then we inject the proteins are specific to cancerous tissue or the tissues which we want to remove. So this is like we call it as hyperthermia applications. And also this is okay. So whatever I have shown the feasibilities. So the interface is open. We can add any specific targets to the interface, and then we can study the ions.
Any role in it?
Sorry.
Hydrogen bonding. What is that? Hydrogen bonding. Over
Okay.
in the is it intervene or influences the?
Yes, it does. It depends on the system which we start with. Yes, it has influence. Of course.
Especially in the pico-Newton force. Pico-Newton force only for the nanoparticles. It is applicable to nanoparticle droplets or what is that role, Professor? Why you are pronounced pico-Newton force?
Okay, so the pico-Newton force is this is the limitation of our technique, because now whatever the pico-Newton force which we plot is the limitation of the magnetic field we apply and the magnetization of the system. So we cannot go higher, because essentially we extract the force from the balancing equation, right? So we balance the repulsive force by external attractive force. So the limit here is using this magnetic changing technique, we can probe this pico-Newton range. That is the limitation of this technique.
Oh, nice, Professor. Excellent. So your research that definitely it brings a new revolution in the data of, especially in the dairy product, protein related the product expiry date may extend or better use. I
Yes,
hope
yes.
there is society related work as well as it will bring the new revolution, Professor. Audience,
So
still now open.
I see there are two questions here.
Yes, bro.
So Venkatesh Priya asked, why choose SDS as a capping agent? So as I mentioned earlier, this is oil water interface. You can put SDS, you can put CTAP, you can put protein, you can put polymers, polyelectrolyte, or even you can put particles. You can put particle at the interface. So the idea of using capping agent is to make the system as kinetically stable, and this model system offers you the freedom of using different stabilizers. That's what I said. The applications are wide open. It's unlimited application. So the interface is open for any kind of tag, right? And the second question is, what about the stability of magnetic microemulsion? Okay, this is a nano emulsion, because the droplet is something like 100 to 150 nanometer, and these are nano emulsions. And yeah, so I started using this material from 2010, and it is 23. So 13 years still, samples are still working. So yeah, this is prepared like 13 years ago. It's still functional. So the stability depends on how proper you functionalize, they were starting material, right? And we have we have optimized what is the concentration of stabilizer should be used for long-term stability. And yeah, this is the example. I'm having a 13 years old sample still with me, and it's working. Is it possible to study interface interaction between solid gas and liquid gas?